Analyzing DNA sequences. And first topic is DNA sequencing, benefits and applications. DNA sequencing is the process to determine the order of nucleotide bases in the genomic DNA or the segment of DNA. And it may involve to know the order of nucleotide bases in any gene and even the larger DNA fragments. In addition to that, it also deals to know the sequence of whole chromosomes. And nowadays, there are different organisms, starting from prokaryotes like bacteria to higher organisms like the humans. At higher rank, as I quoted the example of human, the whole genome sequence, it is now known. The sequencing of DNA first time accomplished in early 1970s when some academic researchers were able to get the sequence of the DNA by using two-dimensional chromatography. इसके बाद with the passage of time जब नई टेक्निक्स जो हैं डीएनए सीक्वेंसिंग के लिए वो मुतारफ़ हुई, तो जो डीएनए सीक्वेंसिंग का जो डाटा है ये एक्यूमुलेट होना शुरू हुआ, तो साइंटिस्ट ने इस डाटा को यूज़ किया within the different disciplines of the life sciences. And these disciplines, they may be in the field of molecular biology, and in molecular biology, it mainly deals to know the sequence of whole genome or any particular gene and the proteins that they encode. So the knowledge that is obtained from the DNA sequences, it can be used to know some mutations in the gene and how these genes, they are associated with particular diseases and even to phenotypes. So this was the basic use in molecular biology. इसके अलावा DNA sequencing को यूज़ किया गया जो evolutionary relation जो हैं उनको find out करने के लिए. Evolutionary relationship means that how microbes they resemble or differ from each other and how they evolved during the course of evolution. दूसरे अल्फाज में हम ये कह सकते हैं कि evolutionary biology का जो subject है, where if we have the idea of the sequences of a particular gene, then it can be used in taxonomy. Taxonomy means that to separate organism into different taxa or rank. For example, in case of microbiology, a particular segment of DNA, which is called as 16S ribosomal DNA, it has been used to find the evolutionary relationship between different groupings of microorganisms. So in this case, the DNA sequencing, it can be used to find evolutionary relationship among a different grouping of organisms. In addition to that, the sequencing data, it is very valuable in the field of metagenomics. Sometimes microbes, they cannot be cultivated in the laboratory by using general culture media. So under such circumstances, microbes, they are analyzed in the natural samples or in the environmental samples. And for this purpose, the field of microbiology, you can say that which is used, it is the metagenomics, where the microbial population, they are assessed within a microbiome or a particular ecological niche. So this is another application where microbial presence in the environment, it can be evaluated by extracting the DNA from the environmental samples, then one can sequence it, and then microbial diversity, it can be evaluated. Then its use in medicine, it is well established, especially to elucidate the role of some genes in genetic disorder. And we can analyze that either some gene is mutated or not. If genetic order it is present, and which particular sequence it is involved in this genetic or particular genetic disorder. And finally, one can also find its application in the field of forensic biology, where information scientifically it can be gathered and examined, for instance from some crime scene. So we can analyze the biological data like blood sample if obtained from the crime scene, and then it can be sequenced, and then further it can be examined to find trace evidence. So these are the different applications of DNA sequencing, its benefits and application. As I told you that DNA sequencing was first time performed in early 1970s when some scientists introduced chemical methods for DNA sequencing, then that were replaced by some enzymatic method, and finally automated sequencing was introduced. And we will discuss all these methods one by one.